Thanks for the invitation. It's really nice to be here. So one of the things that I've appreciated following some of the work coming out of Arena is that you guys are really good at combining disciplinary methods kind of across systems. And so hopefully, so sometimes when I give this talk, it comes across a little bit crazy. Hopefully you guys are kind of used to this approach of looking across systems. And the argument I'm going to make is that some of the kind of rules and logic that applies to social interactions and other organisms also carries over very nicely to viruses. And I'm going to try and illustrate why that can be helpful. Okay, so first of all, what the hell am I talking about with viral sociality? So the key thing is to really zoom in and think about what viruses need to do. So all viruses are basically trying to do two things on a molecular level. They're trying to replicate their genomes, and they're trying to package them inside virions to infect a new cell. And the special thing about both of these processes is that they require gene products that can typically be shared. So typically within an infected cell, a replicase enzyme that replicates one virus can also replicate other viral genomes, provided they've got the right packaging signals on the end. So some outcomes of this can be beneficial from the viral perspective. You can have kind of like feedback loops within the cell, that kind of thing. But this can also lead to a downside, because every time something is shared, it can also be stolen. And we've previously argued that cheats and viruses are kind of quite similar to cheats in other organisms that you might be more familiar with. But one of the key differences is that cheating and viruses are very, very common. And the reason why is because on a kind of physical sense, cheating is very, very straightforward in viruses. So if we have this shared pool of shared gene products, replicases, capsids, etc., to be a cheat in a virus, all you have to do is delete the section of your genome that codes for that gene, and then you're shorter, which gets you an advantage. So you're no longer producing the gene that everybody needs inside the cell, but you're getting kind of disproportionate advantage because you're shorter and can be replicated at their expense. So when I say the word cheat, kind of the rest of this talk, what I'm referring to are these shorter versions of viral genomes that are not producing something that's needed, and instead they're stealing it from the full-length cooperative viruses. So my background is in social evolution, but something that stands out is that when you start to look at the history of virology, it turns out we've always known about these kinds of social interactions. Kind of right from the earliest days of virology, we've been aware that viruses don't just infect a cell on their own, but actually interactions between them at the molecular level are very, very important. You kind of can't help but see them. So a lot of my previous work has been trying to argue that we can take these kind of mechanistic examples, we can draw analogies to evolutionary biology from elsewhere, and that can help us to explain evolution of viruses on a macro scale, things like new types of genome organization, that kind of thing. And this also raises a lot of new questions. So there's a lot of questions about kind of things that viruses can teach us that we wouldn't think about from social evolution more broadly, or open questions about viral traits that kind of seem particularly puzzling. But I'm not really going to talk about those today. Instead, I want to focus today's talk on the consequences of viral sociality, not on a macro scale, but on a more micro scale, and how they might matter for the dynamics of a specific viral infection. So I threw up this little timeline earlier where we kind of look over the history of virology, and we can see that social interactions have kind of always been understood to be important. One of the really key moments here is that this kind of circle in 1947, which is the first time the influenza virus has been passaged in the lab. This is Preben von Magnus. And one of the kind of phenomena that was noticed was that every time you try to passage flu in the lab, you get these cycles. So the population goes up and down. And it turns out that these cycles are emerging because you have deletion mutants emerge of flu that knock out one of the three replicase genes, so PA, PB1, or PB2. They can accumulate, that crushes the population, and then there's kind of cycles. So these were called von Magnus cycles, later known as defective interfering genomes, and more broadly, we think they're a specific example of a type of cheat in viruses. Now, we see these guys everywhere in the lab. Basically, every time you try and grow, particularly animal and plant viruses, you find these guys accumulate unless you bottleneck the population to reduce co-infection. And the ubiquity of these guys meant that in the kind of original review that really coined the term defective interfering genome from Wang and Baltimore in 1970, there was this idea put forward that if every time you try and grow a virus, these cheats accumulate and crash the population, maybe that's happening in nature as well. Maybe viral infections have a degree of being self-limiting, whereby if the infection takes off, you have lots of virus that creates conditions under which it makes sense, it's evolutionarily favored to drop shared genes and exploit full-length viruses. So this is kind of like an open question. And until recently, it's been quite hard to answer this because we've not had sequencing data. Now, I'm not going to go into the details of exactly how we detect these because I don't really have time. But the data I'm about to show you comes from a collaboration with the Wadsworth Center at the New York State Health Department and with Andrew Routh, a collaborator of mine who has a kind of a library prep method that's very good for detecting viral cheats. And what we've done is we've looked across a couple of hundred naturally infected influenza patients. And on the x-axis, we've got each patient. On the y, I've just plotted the abundance of the most abundant kind of big deletion in a polymerase mutant. And the key thing here is that these flu cheats are common. So this is not something where they're persisting at, like a small percentage of the population. This seems to be a phenomenon whereby these cheats are persisting at appreciable frequencies in these natural infections. So these are not samples that's been passaged in the lab at all. These are just purely taken straight from a swab. So this is kind of the empirical pattern, that these cheats are out there. And I think the question that really gets raised is, okay, these guys are out there. We know in the lab they can crush the infection. What are they doing? What are the consequences of this? So for the kind of second half of this talk, I'm going to talk about our kind of approach to model the potential outcomes of these dynamics and try to connect these to patterns in data. So you don't have to write too much about the details of this, but we basically have a stochastic birth-death model where we're tracking the population of deletion mutants within a host. And we're fixing this to what it looks like for the first 3 days of an influenza infection. And what I'm going to show you are the dynamics of this when we allow a little bit of cheating. So when we allow shorter viral genomes to have a kind of selective advantage in co-infection. When this happens, we can see that we start with a world where everybody's a cooperator, a full-length genome. And as you kind of play the infection out, if you can get mutations that kind of delete the sections of the polymerase genome, and that gives you an advantage, you can get these kind of sequential cheats invade. Shorter viral genomes start to invade and sweep in the population. So this broadly recapitulates what we see in the lab, but importantly, we have a stochastic model here. So we can replay these dynamics with exactly the same initial conditions and parameter values. And what we find is that sometimes the viral population kind of like persists if frequency dependence at an equilibrium, like on the left. But sometimes if you get a really good cheat early enough in the infection, we predict extinction. So the whole population gets taken over by these cheats that are not producing the essential public goods. And if this happens, the infection is always going to go extinct. And it kind of comes down to this question of do you get a good cheat early enough? So what this means is if we think back to our original kind of idea from the 1970 paper that maybe viral infections are self-limiting, what we're finding is that a fairly minimal model without invoking anything particularly clever that looks broadly like what we see in the lab does suggest that sometimes you might get not just these cheats spreading in a viral infection like we see, but it might suggest that actually sometimes they might fully drive extinction of the population. So what we're doing with this model is we're kind of exploring, we're parameter sweeping kind of different variables. So here we've got noise and cheat advantage, and we're saying how often do you get the kind of outcome on the right where you have extinction versus how often do you get the outcome on the left where the cheats spread but they don't quite go extinct. And this means we can kind of explore various properties of viruses to start to make broad scale predictions. So for example, we can ask about the importance of intrinsic demographic noise in this process, or we can ask about mutation rates. So we seem to find that higher mutation rates mean you get a better cheat earlier, so you're more likely to go extinct, but actually the details of that mutational process are less important. And the kind of future direction I want to go in this model to just briefly mention is one of the advantages of casting this model in a way that predicts deletion sizes is in principle, I think that we can take these model dynamics, we can infer kind of population level properties of kind of diversity in the cheat population. And I think we can link these to patterns that we see in data to kind of go beyond this kind of heuristic description and actually quantitatively start to infer these cheats that we see in the lab, how much of an advantage do they get in nature? And perhaps most importantly, are there properties of these kind of selective sweeps that we're seeing of cheats that might mean that we could take a measurement at an early time point of infection and predict whether that infected patient is en route to cheat extinction or not cheat extinction? Okay, so I've gone reasonably fast because I wanted to give a little bit of an overview of kind of the data side, the quantitative modeling kind of together. But broadly speaking, I think there's three things and I also want to leave plenty of time for questions. So I'm happy to talk about more of these things in detail. But in general, I think there are three things that I want to kind of emphasize. The first is that because of the way that viral gene products can be shared within cells, sociality is not something that's like optional for viruses. It's kind of intrinsic to the way that they replicate. What this means is we can draw analogies between this process and other examples of evolution that we understand quite well, and that can help us to kind of better understand these broad scale patterns. And what I've talked about today is my attempt to go beyond that and try to say not just broad scale patterns, but actually can we use these social interactions to start to understand dynamics at the level of the individual infected patient. And I focused on one specific outcome, which is extinction of the viral population, because that's the most dramatic, right? That's the most in your face. It's the one that most clearly links to things we care about, such as clinical outcomes. But in general, if these things can be at 50% relative abundance, and we know they have strong phenotypic effects, I think there are good theoretical reasons to think that viral cheats could be playing an important role in other things we care about, such as cross-species transmission or evolvability of viral populations. So I'm interested in kind of interacting with people who have data sets, if you might be interested about working on this kind of problem. Okay, so the modeling I showed you comes from a collaboration with some collaborators at Santa Fe. The data is from a Wadsworth Center in New York City. And then the final thing is I'd like to say that I'm opening my lab at UBC next year. So I'm recruiting and looking to talk to people who are interested in this kind of thing. Thank you.